begin with a really general oversimplification of sensory systems and how they're organized. Okay, pointing out that there's this sort of general stream of processes that have to occur, right? You've got some kind of external reality. You have to have a receptor that can detect that external reality. You've got to transduce that into a neural signal. You've got to be able to propagate it, modify it, get it to places in the nervous system where you can become aware of it, you can perceive that. And then there is feedback that leads to that modification. So as we go through sensory systems and we talk about the anatomy of them and relating that to function, keep that general stream of processes in mind. Okay. And also when we're thinking about sensory systems, some of the issues that you have to keep in mind are things like the specificity of the receptor to the particular types of stimuli. You need to think about mapping. How sensory space is mapped is a really big deal. Laterality and crossing of information. Hopefully you already get the crossing thing is really, really important. You're going to have to keep up with that. Understand that there are distinct sort of sub-pathways within each sensory system, and they are important to understand the differences in those pathways. And as we started talking about on Wednesday, there are primary areas of the central nervous system associated with the sensory system, and then there are association areas. And so we'll keep those designations in mind as well. Okay, so we're talking about vision today, and so we're going to start with how we receive that visual information, right? So we've got this world out here, and somehow we have to detect that external reality, right? And that's all going to start in the eye. So this is just a drawing that illustrates the main parts of the eye. Now there's going to be more parts that are labeled on the picture in your book than what I'm going to talk about. But we're just going to highlight some of them now. So understand that when light comes into the eye, right, so out here coming into the eye, you're going to end up getting a image of visual space on the neural retina that's in the back, so right here, that's going to get inverted and reversed by the lens. Okay, and the inverted part, the reverse part, you got to really keep this in mind. Okay, this is going to be really important when you're trying to map visual space to the surface of the retina and understand where it goes. Okay. So, parts we want to talk about, I want to mention the cornea, okay, which is a transparent connective tissue that is non-vascular. Right? Light's got to pass through that. Once it goes through the cornea, it's going to enter into the anterior chamber. Okay, so that anterior chamber is in here. Okay, and then you have the iris located here. That makes up the posterior part of that anterior chamber. In addition to connective tissue, there are a couple of muscle groups you should know about. The sphincter muscles that are concentrically arranged, right, and then the dilator muscles that are radially arranged. Okay, and then the space between that is the pupil. Okay, so then from the iris going back, we have this small space, that's the posterior chamber. And then the lens, and the lens is held in place by these suspensitory ligaments that are called zonial fibers. Okay, and then we have the vitreous body behind the lens, between the lens and the neural retina. Okay, so the retina has two parts. Right, there is a neural retina component, and there is a retinal pigmented epithelium. We'll get into that more in a moment. And then I want to point out the optic nerve. Okay, that is here at the most posterior extent of the eye. Here's the optic nerve. You can see that there's dura surrounding the nerve, so there's subarachnoid space. Here's the nerve, the axons coming from the neural retina entering in here. And there's the central retinal artery that's coming in to supply the inner part of the neural retina. And then there's also ciliary artery here that's coming in to supply the choroid in the outer part of the retina. Okay, so I mentioned to you those anterior and posterior chambers. I want to point out that there is a fluid that's produced by the ciliary body, okay, which is the muscle here and the epithelial tissue lying over it. And that epithelial tissue is producing this fluid that fills the posterior chambers, and it flows forward through the pupil into the anterior chamber, and then is drained out through canals located here on either side called the canal Schlemm. And so if anything would block that canal, you would have an excess amount of fluid building up. Okay, and then an example of that clinically is cases of glaucoma involve elevated fluid. Not saying that blocking this causes that, but understand when you have elevated fluid, one of the situations you can have is glaucoma. And also understand that this fluid that's produced here in the posterior chamber, yes, it flows forward and drains out here, but understand that these suspensitory ligaments that are holding the lens in place, they're not like a wall, they're like strips. So there's space between them, so that fluid is also able to flow back here into the vitreous as well. When we talk about the retina, like I said, there really are two parts of it. There is the neural retina and the retinal pigmented epithelium. Okay, and so in this picture that you see, okay, orient yourself. This is the vitreous here. Okay, and then this is going to, here's the choroid. So this is going to be back, right, the most posterior part of the eye. This is going to be going anterior, going toward the lens, coming forward. Okay, so when light is coming in, right, light is coming from here, going this way. Okay, that's important. You're going to need to really keep that in mind. The path of light is going to be opposite of the path of information flowing back out of the eye. Okay, so the retinal pigmented epithelium are cuboidal epithelial cells here in the back that sit right up against the choroid. And you can see how they kind of interdigitate into the outer segments of the photoreceptors. Okay, so that's our outermost part. So then we go into the neural retina, and we have a series of layers, basically 7 layers, that I like to think of it as kind of alternating like a sandwich. Okay, in that you've got the outer and inner segments. It's going to compose a layer, and then you've got this alternating pattern of a nuclear layer, so where you've got cell bodies, and then a plexiform layer where you've got synaptic connections, and then another layer of cell bodies, another layer of synaptic connections, another layer of cell bodies, right? And then the output layer here, the nerve fiber layer. Okay, so I kind of think about it as like I've got my bread here and here, and then I've got this alternating pattern of lunch meat and cheese, just kind of stacking on top of each other. Okay, that sticks in my head. I don't know whether that's going to make you hungry or not, want to think about the eye, but that's how I remember that there's this sort of alternating pattern in place. So what I mentioned, I want to emphasize again, light is coming from here, and it has to pass through all of these layers of the neural retina in order to reach the photoreceptors to the outer segments, which is where you're actually going to get the phototransduction response to take place. Okay? And then that's going to initiate a signaling pathway that's going to go this way until you get the signal coming out via the axons of the retinal ganglion cells. Yeah?
Do we need to know the identity of the cells that are labeled in letters?
Yeah, so you should know what the main cell types are, right? So, and we're going to talk about them, but I will just highlight them now, right? So you've got photoreceptors, you've got cones and rods, right? Those are your main types of photoreceptors. You've got, then in this inner nuclear layer, you've got the bipolar cells. Okay, but then you've also got horizontal cells. Okay, so they have lateral connections. We're not going to worry about the physiology of why they're important. Okay, so you just need to know that they're there. And then you've got amacrine cells. And so you see the horizontal cells are making synaptic connections between the bipolars and the photoreceptors, and then you've got these amacrine cells whose connections are between the bipolar cells and the retinal ganglion cells. They're modifying the signal. And then we've got the retinal ganglion cells here, whose axons form cranial nerve 2, okay, are projecting out of the eye. So photoreceptors, I know you've heard of rods and cones, right? I just want to give you a quick reminder of some of the important things about them. Understanding, obviously, that the shape is different, right? Rod shaped versus cone shaped here. They use different molecules to do their phototransduction. The key things to really think about are the rods are far more sensitive to light than cones are. Okay, so what that means is that when you're stumbling around in the dark, your rods are going to do a better job of helping you see than your cones are. Your cones need more light in order to allow you to see. Okay, so that's what this means, right? This is sort of your night vision. Okay. And there's about 100 million of these guys. What you should also note is that these guys, you're going to see this later, but they're located more peripherally in the retina. Okay, and so then we've got cones. They are specialized for different wavelengths, right? So you've heard all about different cones for different colors that creates color vision. Okay. And what I want you to note about cones is that they tend to be located more centrally in the retina. Okay, so this is a heat map that shows you the distribution of rods and cones on the retina surface, okay, as if it was sort of laid out flat. You should see that it's a heterogeneous distribution, right? They are not mixed evenly, right? So when you look at some of the drawings, we put rods and cones side by side and kind of intermix them because we're trying to show you the different types of photoreceptors. But what I want you to recognize, right, is that they're not intermixed all over the retina. Okay, there is a general pattern of distribution where, okay, and with a heat map, like blue means cool, there's less, right? And we go toward oranges, reds, that means there's more. Okay, and so what you see is that in the peripheral parts of the retina, you have more cones. You have more rods, right? And less rods in the central part. Conversely, you have a concentration of cones here in this very, very central part of the retina, and you don't see cones out here in the peripheral part of the retina. Yeah.
You said that they use different molecules for signal transduction. Do we need to know the pathways for those signal transductions?
So we are not going to worry about the signal transduction pathway. Okay, so there was this spot that I was pointing out to you where we have this high concentration of cones. So in the posterior part of the retina, in the central area, it's called the macula. And in this macula area, we have this high concentration of cones. And in the very center of the macula, we have the fovea. Okay, and so the fovea is this depression. So you can see it nice in this SEM image. So you see this large depression right here, right? The surface is kind of coming down, and then there's this big sort of cliff that it drops off right down into this foveal depression. And you can see that here in this cross section, also looking at the retina, where there's this big dip down here. This area, the fovea, is specialized for high visual acuity. Okay, so what happens where you get this dip is that all these cell layers that you have out more laterally, when you get here, the fovea, they're pushed to the side, right? So that the only layers that are here are the outer and inner segments of the photoreceptors and the outer nuclear layer. So the plexiform layer, the internuclear layer, interplexiform layer, retinal ganglion cells, all that's shifted. Why would you do that? How does that relate to acuity?
It transducts the signal faster as opposed to having to interact with all the other cell layers.
Well, no, the signal's still going to have to kind of go and interact. It's just that everything kind of got pushed sideways.
So it could be more sensitive on those cells. We have those layers moved out of the way.
Right, so one thing is, it increases your sensitivity. So remember, light's coming from out here, and it's got to pass through all those layers to get to here, where you actually have the phototransduction happen. Okay? So if you got a lot of stuff in the way, the photons are going to get scattered, they may get absorbed by other things, and so that's going to reduce your sensitivity as well as the acuity of what you see. So if you move all that stuff out of the way so you have a direct path right to the sensor, you end up getting more sensitivity, better acuity. All right, so then I mentioned we have our inner nuclear layer. That's where we have the bipolar cells, the horizontal cell bodies, amacrine cell bodies. Okay, and then we've got another plexiform layer, and then we have the ganglion cell layer. Okay, so these are the retinal ganglion cells, which are the cells that project out. Generally speaking, when we're thinking about the image-forming retinal ganglion cells, you can think of them as being two main types. Okay, there are the alpha cells, which I've also indicated here are sometimes called M cells. And you can see the big difference between them and the beta cells, or the P cells. Okay, so there is a much greater dendritic arborization on the alpha cells compared to the beta cells. Okay, the cell bodies are different in size, and where they're getting their inputs from is different. And this is what really matters in understanding ultimately sort of the pathways that these are going to contribute to. So you have your alpha or M cells, and they mainly get their input from rods. And remember, where are rods predominantly found?
Periphery.
In the periphery, right? Okay, and so they are, they're good for detecting motion in particular. Okay, motion, contrast, those kind of things. Your beta cells, or P cells, okay, they are located more in the central retina, right? Their input's coming from cones. Okay, and so your beta or P cells are going to be good for color, right? They're going to be good for high acuity and color vision. Okay, so what you should begin noticing is that we're kind of taking visual information and we're separating it into some different anatomical groups that are going to lead to some of those distinct functional pathways within the visual pathway. Okay, so those retinal ganglion cells, their axons are going to form that nerve fiber layer that is the innermost layer of the retina, of the neural retina. And those axons are going to all travel posteriorly to converge at the optic disc. Okay, and so this cross-sectional view is just showing you that nerve fiber layer and those axons going down here to form the optic nerve. And so remember, I mentioned that central retinal artery that's also coming in through the optic nerve, right? It's coming in through this way. And this is a picture that's sort of from the inside of the eye that allows you to see how that central retinal artery comes in at the area of the optic disc and then branches out to supply blood to the inner parts of the neural retina. Okay, and so the optic disc, you guys have done the little experiment, where you like hold your thumb out of a certain position and, your thumb disappears. You ever done that? Right, so you know that that's your blind spot, right? That's that place where you can't see because there are no photoreceptors right there, right? There's no photoreceptors because that's where the axons are exiting out. And so if you were to lay out visual space on a coordinate kind of drawing like this, and you're going to see these over and over, then what you'll notice is the fovea, remember that's where all the cones are clustered, we have our color, high acuity vision, is in the center, and then the optic disc is located there just a little bit laterally to the position of the fovea. All right, so we've got these axons. They have headed out from the optic disc to form the optic nerve. Okay, and so they are traveling. There's the optic nerve. Okay, and they're coming along here on the inferior surface of the brain, and then they come to the optic chiasm right here. Okay, and you can see the optic chiasm. They're right right anterior to where the hypothalamus is located, so I've tried to kind of block that out so you can see that on this inferior view. And so at the optic chiasm, what happens is you get a partial decussation of the axons. We're going to really get into that in a moment. Okay. And so then the information continues to travel here in the optic tract. See, wrapping around lateral to the hypothalamus, coming around lateral to the crus, okay, coming in to the LGN. And so then I've got the midbrain colored here in yellow. So superior colliculus is in here. There's the medial geniculate sitting here in pulvinar there. Okay, and so then you can also see it in the cross-sectional view here. I couldn't quite get a section that showed you both the tract and the chiasm, but you can see the tract coming in this way and coming around to the LGN. One way to remember too, when you're looking for it, is the hippocampus is usually sitting near where you can see sort of the roll up of the hippocampus. And then you'll sometimes see the lamination of the LGN just anterior to that a little bit. Okay, like I said, at the chiasm, there's a partial decussation. Okay, so that means some axons cross, some don't. And so here we are. Some are going to cross. That means they're coming from the optic nerve here and going this way. And some are in this optic nerve and they're going to stay in this optic tract. All right, what's the rhyme or reason for that partial decussation? Okay. So it relates to understanding visual space. Okay, so here's a drawing where, imagine it's sort of like you're looking down, okay, on the head from above. All right, so this is my right eye then in this drawing. So I've got visual space out here. Okay, and so if that was me right now, right? Again, I've got my left side, which in this drawing is blue, and I've got the right side that's red. Okay? And what you start by noticing, right, is the visual space gets mapped onto the retina in the inverted reverse kind of way. Remember, I mentioned that to you? Okay, so I brought a old soup label. Wine word that I bring. So I want you to really be remembering the fact that the retina is curved. Okay? And so it's this curvature that really, when you remember that, helps you recognize how things are going to get inverted and reversed. Okay? And so in a moment I'm going to try to shine my pointer at it and illustrate it, but it may or may not work. But keep remembering that there's this curvature. Okay, so left, for this eye, right, left visual space over here, okay, that's going to be the nasal hemifield in the right eye, and that's going to map over here to the temporal retina, okay, of the right eye. Well, you can see then this nasal hemifield that's left that maps over here to the temporal retina. Follow the blue line. See that those axons remain ipsilateral. Okay, so temporal retinal axons always stay ipsilateral. Okay. And so now the right part of visual space, that's the temporal hemifield in the right eye. Okay, and so that is mapping into the nasal part of my retina, right? Because my nose is here, right? So this is the nasal part of my retina. And those axons, the axons from the nasal retina, they always cross. Okay, so the partial decussation is that temporal retinal ganglion cell axons remain ipsilateral, nasal retinal ganglion cell axons cross. All right, why do they do this? Okay, so I think that to help you understand why they do it, you need to understand how visual space is represented on the retina, and then it's going to make sense why we have this partial decussation. Okay, so first of all, you need to understand that most of visual space is represented in both eyes, right? And if I cover one eye, I can still see most of the classroom, right? There are regions that I can't see, right? If I cover my right eye, there's parts over here I can't see, correct? And so that would be this monocular zone, right? And so there's monocular zones on the lateral extremes of your visual fields. Okay, but the bulk of your visual space in the middle is binocular visual space. So both eyes are seeing that space. Okay? And so that lateral part of visual space, okay, that is monocular, just remembering that curvature, okay, that's always going to be coming in onto your nasal retinas. Okay, so the lateral parts are coming nasal, and the more medial positions are going to go to the temporal parts of your retina. And so like when you're taking the test, and I see you guys going woo, woo, like this, right? I know you're working on a vision question. Okay? Yeah.
Can I ask you a question about the last slide?
Well, we'll see.
So for the left eye, so the temporal retina cross.
So yeah, so over here on the left, I follow the red line. Okay, so here we are, temporal retina coming down, stays ipsilateral, right? Temporal retina always stays ipsilateral, nasal retina always crosses. I'm going to explain why in just a minute. Okay, I'm getting you to an idea. Okay, or I'm trying to anyway. I hope I am. All right. Okay, so just to make sure we're all understanding these drawings. Okay, see, you're working on it right there. I just see you guys doing this kind of stuff. All right, this is. If you're going to look and try to understand visual fields, okay, you're going to do it with these coordinate plots. Okay, and so you're going to have the left eye and the right eye's plot, right? So this is the fovea and the center. Okay, and you're going to take these plots and you're going to map them. You're going to divide them up into hemifields and quadrants. All right, so kind of using the fovea as your dividing point, right? So things that are moving lateral from the fovea, okay, are going to be in your temporal hemifield. So this is the temporal hemifield of the left eye, this will be the temporal hemifield of the right eye. Okay, so the temporal hemifields are out here, right? And the nasal hemifield is the medial, to the fovea. Okay, and then you also divide into quadrants. Okay? And so you have quadrants up here, superior, whether I mislabeled that in a second, and inferior. So what you've got is space, superior visual space, it's going to be mapped on the inferior surface of the retina. Okay? So let's go to that. We'll go to that. Okay, so in looking at visual space then, remember we have the binocular zone. So this blue circle is left visual space. This dividing line is left visual space from right visual space. And so the left eye, what does the left eye see? Well, the left eye sees part monocular, part binocular, and it sees some of the right side. So that's why my drawing, I'm divided in half here. All right, so this temporal hemifield and nasal hemifield. All right, so I told you about that. So this temporal hemifield projects to the nasal retina. Right? Nasal hemifield projects to the temporal retina. That's that crossing. That's where you have to keep this in mind. Okay? If I want to try to get something, let's say that we're going to say that this is the nasal part, so my nose is the handle here. Okay? If I want to try to get light onto this nasal part of the retina, can I shine light from over here and get it to hit it? It doesn't work, right? I mean, if I try to, I can't do it. I can make it hit over here on the temporal, but I can't make it hit there, right? I have to come at it from the other side. Okay, so that's what's going on here, right? That's why you have that crossing going on, because of the curved shape of the retina. Okay, so both eyes have this pattern of seeing a little bit of left and a little bit of right visual space. So left visual space, that's the blue color, is seen in both the temporal hemifield of the left eye and the nasal hemifield of the right eye. Okay, so I got information about you guys sitting over there. I've got that on my nasal retina here and my temporal retina here. I need to bring that together. Okay, Okay, otherwise I've got two separate maps, okay? And so that's what the partial decussation does. It brings together the information about the same spot, brings it all to the same side of the brain. And because, remember, so my blue line, my blue space here, left visual space, is nasal retina of the left eye, temporal retina of the right eye. So the nasal crosses, so my blue from here is coming this way, and my temporal stays ipsilateral, so it's staying on that side. Now that I've crossed the chiasm, I don't have a mix of blue and red anymore on one side, do I? I've got all blue over here, and I've got all red over here. Okay, so that partial decussation allows me to bring all the left side of the world to the right side of my brain, and bring all the right side of the world to the left side of my brain. Okay, so now that I've brought all of, let's say, left visual space across the chiasm, so it's all over here in the right optic tract, okay, and so then it's going to go to the lateral geniculate nucleus. Okay. And so the lateral geniculate nucleus is going to get axons that are coming from the ipsilateral eye and the contralateral eye. Well, those inputs segregate out into layers. So you have this pattern of inputs of C means contralateral eye and I means ipsilateral eye. So I've got all of left visual space coming into the right LGN, and depending on whether it came from my ipsilateral eye or contralateral eye, it's going to go to different layers in the LGN. Okay, so ipsilateral inputs, so coming from temporal retina, are going to layers 2, 4, and 6. 2, 3, and 5, my bad. 2, 3, and 5. And then, so contralateral from nasal retina of the other eye is going to go to 1, 4, and 6. Okay, there should be another thing that you notice here, okay, and that is that there's kind of a different color of these guys versus these layers, right? What's that all about? Okay, well, there's another level of separation, and that is whether or not the axons are coming from those alpha or M cells, or whether those coming from the beta or B cells. Okay, so we separate input from the M cells, go to layer 1 or 2. Input from the P cells, go to layers 3 through 6. Okay, you remember what are the alpha cells, what are they good at? Movement, motion, yeah, exactly. So I've separated movement and motion information from color and acuity information in the different layers of the LGN. All right, so we did that. Okay, so now that I've gotten this information to the LGN, so LGN has visual information from the contralateral visual space, right? That's going to then project ipsilaterally to primary visual cortex. Okay, so here's my LGN. And so then those projections, those axons are going to travel in the optic radiations, right, that's in the retroreticular limb of the internal capsule. It's going to come back here to the occipital lobe. Specifically, it's coming back to right here, this dotted line is showing you the calcareous sulcus. Okay, and it's going to primary visual cortex, which is located on the banks of the calcareous sulcus. Okay, so cuneus is above the sulcus and the lingual gyrus is below the sulcus. So primary visual cortex is located right in here. Okay, I mentioned to you about the division of visual space into fields and quadrants. Okay, and so the superior quadrant, so I had messed that up on that other slide, I'm sorry. The superior quadrant of visual space out here is going to be on my inferior retina. Okay? And so if we were to look here, I blocked in one of these quadrants, right? So this is my right eye. What hemifield is it? nasal, so it's my nasal hemifield, okay? And so that is superior visual space, so that's going to be where my retina? Inferior, okay, so it's going to be on the inferior temporal part of my retina because it's up here in the superior nasal hemifield space. Okay? So where are those axons going to go at the chiasm? It's temporal retina, so they're going to stay ipsilateral. Okay, so if this is my right eye, then that information is going to stay on the right side of the brain. It's going to go to the right LGN. Okay, those optic radiations actually separate, and there's two different paths to get to primary visual cortex. Okay, so you saw, I said primary visual cortex was here on the banks, right? So there's up above the calcarine sulcus and below the calcarine sulcus. Okay, so there's a separation based on superior and inferior visual space. So superior retina, okay, that's coming in, you see the green line here, so inferior visual space, superior retina. Those projections are going to take the superior path in the optic radiations back to primary visual cortex. So that is different than from superior visual space, that's going to be on the inferior retina. Okay, inferior retina is going to take a different path. Okay, this is Meyer's loop. So these axons, they go kind of rostral first, curve around the lateral ventricle, and then head back, and they come in on the inferior bank of the calcarine sulcus. Okay, so now I've separated superior and inferior visual space, and since I'm coming out of LGN, I've already separated hemifields as well. Okay. I also want to point out to you, in terms of where these axons go into visual cortex, this black line is representing the macula, right? So that's that area in central retina where you the fovea is within it. That's where you've got your central vision. That makes up only about 10 degrees of visual space, but it's where most of your photoreceptors are found. And so it's going to take up a large territory. Remember in the homunculus, your face, your fingertips, they disproportionately took up a large amount of space in primary somatosensory cortex. Same deal here. Your central visual space takes up a disproportionately large amount of territory in primary visual cortex. Okay, and so you can see that here, the size of the area that's getting information from the macula is larger than the size of the primary visual cortex that's getting information from the periphery. Okay, so remember those alpha cells and beta cells, right? Magnocellular and parvocellular. Alpha or magnocellular, good for motion, input from rods, comes into layers 1 and 2 of LGN. Once the information gets back to primary visual cortex, which is area 17, okay, and so you can see that here, so this pink color, okay, this is on that medial view, right, and it wraps around just a little bit of the lateral view. Then you get areas 18 and 19, so these are secondary visual cortex. So this is getting into association. Remember I think you learned about primary and association. On Wednesday, these are unimodal association areas. So they're doing just vision, but it's not primary visual cortex. And then you move into multimodal association areas as you send that visual information out further. And that visual information, we describe it as following dorsal streams and ventral streams. Okay, so the dorsal stream of visual processing, right, it's gonna come up this way, right? And so it's gonna include this area, 7 that's shown there. Okay, so that's gonna be that dorsal stream. That is where that magnocellular pathway is going. Okay, so that pathway that's really good at motion, space, those kind of things. Whereas the parvocellular pathway, remember that's primarily your cones and your acuity, that's following this ventral stream. So that's coming down here into the temporal lobe, inferior temporal lobe areas. And so this is where you're going to have processing related to color and form and acuity. I just want to give you an interesting example of a story of a woman who had strokes that impacted ventral stream predominantly. Okay, so I'm giving you the answer here. What happened was, sorry, my bad, not the ventral stream, that's what was preserved, that impacted her dorsal stream. Okay, and so she could see, she had color vision, she could read, right? So her acuity and color, all that were fine. Okay, but she had a lot of trouble with visual motion. Okay, so it's this dorsal stream, magnocellular pathway. What would that be like to have trouble processing that dorsal stream information? Okay, these are examples that she told about. She couldn't pour into a cup because she couldn't perceive the motion of the fluid moving. Right, it kind of was frozen to her. She couldn't tell as it was gradually emptying out and filling up. She didn't feel really good in a room with a lot of people because she couldn't tell people were moving. It was like they were in a spot, and then all of a sudden they'd be at another spot, and she'd be like, "How'd you just get there?" Because she couldn't perceive the motion. She couldn't cross the road because she couldn't perceive the motion of cars coming down the street. Right? She could see them all, but she couldn't perceive that motion processing. So recognize that you get unique deficits to tell you whether or not it's related to magnocellular, dorsal stream, motion processing, or color vision, and form color processing. Okay. Yeah.
Does that also affect your own proprioception?
What's that?
Does that also affect her own proprioception?
Her own proprioception. She didn't report any issues with somatosensory aspects of motion. She could still detect other, inputs coming in, like she could tell when something moved across her arm, those kind of things. So I don't think it would have affected that as well. It didn't specifically speak to it.
Did she tell she was moving when she walked?
Yes. She just couldn't perceive outside visual objects moving. Okay, so if you want to look at somebody who's having a visual problem, you want to try to map that, okay? And so remember these coordinate drawings that I was mentioning to you, right? You're going to map those drawings by, you're going to have somebody cover one eye, and you're going to figure out what they can see, right? And the areas that you can't see, whatever degrees of visual space that is, you're going to indicate that with darker colors to say this is the area of deficit. Okay? And you're going to do the same thing with the other eye. Okay, and so this is just an example, this comes from your book, of a deficit that occurred where most of the left eye's visual field was lost, right? And then this particular part of the right eye. So, where is that on the retina and in the visual pathway?
The visual pathway that.
I just heard it once. Okay, inferior nasal. Okay, inferior nasal retina is where that part of visual space is going to be. So where are those axons going to do at the chiasm? They're going to cross. Okay, so that's nice because if I've got a problem over here that affects like almost all of my left eye, it would be really confusing then if I had a lesion like in here, because that would not be joining together. Okay, so where could you imagine nasal retinal axons from one eye and all the axons from the other eye being impacted? Okay, if I'm gonna get both hemifields in one eye, it's gotta be the optic nerve. Okay, because that is pre-crossing. Okay, so that's one clue right there. Okay. In this particular case, like I said, you've got to impact the optic nerve. Okay. And so that's where the lesion site is. So you're impacting that optic nerve, but this is a drawing that illustrates it. What you've also done, though, is the size of in this case, an aneurysm here, is impacting those crossing axons a little bit at the chiasm. And so then that's leading to this deficit area as well. I'm going to skip talking you through this slide, but you should look at it and figure it out on your own. This is helping you figure out how to switch around the orientations. Okay, so you're going to want to think about that and do that. Another practice one. Where do you think this lesion is to get this kind of visual field deficit? It's at the chiasm, right? Because this is temporal hemifield, which means nasal retina, both eyes. Where do you find both of those? At the chiasm. Okay, so that's that deficit right there. Okay, and if you had a deficit like this, so it's just a quadrant. If you have just a quadrant, you should know that you are past the LGN. Okay, you are in optic radiations or impacting just one bank of primary visual cortex on one hemisphere. Okay? Anytime you see just a quadrant, it has to be from the after LGN going back. Okay? And so in this case, left inferior visual space means right hemisphere, superior pathway to cuneus. Okay, there are non-visual pathways as well. So not all the visual input goes to the LGN. There are other structures that receive it. Superior colliculus being one of them. Pretectal nucleus being one of them. Okay, so superior colliculus and pretectal located there. There's superior colliculus, there's pretectal in the drawing. Okay. We've been talking about eye movements, and so I want to hit this for you real quick. How does this work? Cranial nerves 3, 4, and 6 are innervating skeletal muscles that move the eye. Okay, so we're almost out of time, so I have to highlight it for you. The oculomotor nerve, so that's here, helping you move the eye up and down and medially. Okay. The lateral rectus muscle is innervated by the abducens, and so that is just moving your eye laterally. And then the trochlear innervates one muscle, superior oblique, that kind of helps you look sort of down and out. And it's kind of an awkward twist, but if you look at the picture in the book, you can kind of see how that would happen. Understand that we've been talking about the medial longitudinal fasciculus some. One important thing to recognize with that is that the medial longitudinal fasciculus connects projections from the abducens up to the contralateral oculomotor. Okay, because if I want to look right, I need my right eye to go lateral, but I need my left eye to go medial. Okay, and so I need to tell my lateral rectus muscle, and so that's going to be abducens, but I also need to tell the contralateral oculomotor nerve to activate the medial rectus of the other eye. Okay, so that's one thing that the medial longitudinal fasciculus does. Okay. In terms of visceral eye movements, you need to understand how the pupil is adjusted. Right? How do we activate those sphincter muscles or the dilator muscles in order to cause the pupil to constrict or to dilate? Okay, constriction is a parasympathetic response, so it's coming from visceral motor, Enger-Westphal nucleus in cranial nerve 3. And that's innervating these sphincter muscles, and so then that causes your pupil to constrict. You can look at the pupillary light reflex as a good indication of about the function of cranial nerve 3, for example, as well as sympathetic and parasympathetic nervous system function. So you shine light in one eye, and what you should get is a constriction of the pupil in both eyes. And so you have the direct reflex, which means the eye that the light was shined in constricted, and the consensual reflex, which means that the other eye constricts as well. If you shine light in one eye and you don't get both of them constricting, you know there's something wrong. And you can, between going back and forth between the two eyes, you can map it out. Is it the afferent limb? That is it the optic nerve? Is it the efferent limb? That is it cranial nerve 3? And so you can look at this as an example. I'm going to skip through that. Okay, we've talked about blood supply in the past, but just to remind you, the LGN is getting it from the thalamo-geniculate arteries, which are coming from PCA. Okay, and also just remembering the primary visual cortex, located in here, is in the territory of PCA as well. Okay, so what do you think's going on here with this guy?
It's got to be a lesion.
Okay, he has a lesion past the LGN, exactly, because we're at a quadrant, right? Which hemisphere are we looking at? Right hemisphere, because it's left visual space of both eyes. So when you see a drawing like this that shows this area of visual deficit, you automatically start thinking, okay, optic radiation's back to primary visual cortex in the opposite hemisphere. Right, it's where you want to look at for where the problem might be. Okay, in this case, he also had, it was a chronic situation, right? It was like 4 months of time. Okay, so, you got to think about causes that can lead to a chronic problem like this rather than acute. Okay. I picked this one because it's something kind of crazy that you probably would not have thought of, right? Because you're thinking chronic, you're thinking, oh, he's got some kind of tumor growing back in there, right? Okay, so this is an example of a arteriovenous malformation. Okay, so what was happening, so that's this is a picture showing you the arteries. The middle cerebral artery, I'm going to use it just went crazy. Okay, in terms of its growth pattern, and so you have this it looks like spaghetti, right? It shouldn't have that type of path there. Okay, and so what was happening was it was affecting, because of this huge malformation there, it was affecting the superior optic radiations on the way to primary visual cortex. And that's how you were getting that particular deficit. So that's it for today. Remember, no class Monday. Dr. Haynes has said he'll be here tomorrow morning in his office if you want to talk. And we'll see you Wednesday.